All right, here we go. So this is a slide that we left off on. We were talking about stem cells. Traditionally, there's been three categories of stem cells. Totipotent, which can turn into any organ in the body or outside of the body, notably the placenta or umbilical cord. Pluripotent, aka embryonic stem cells, that can turn into any tissue within an adult human body. And then multipotent, aka adult stem cells, which are pre-differentiated or partially differentiated pluripotent stem cells. However, in 2008, a researcher from the great state of Wisconsin, collaborating with a researcher in Japan, came up with a new one, induced pluripotent stem cells. And I personally find IPS stem cells, or induced pluripotent stem cells, to be very exciting. An induced pluripotent stem cell is a multipotent stem cell that has been reprogrammed to act like a pluripotent stem cell. In other words, you get all of the benefits of embryonic stem cells without any of the ethical dilemmas associated with harvesting embryonic stem cells. There is a large portion of the population that is not okay with human embryos being created and destroyed for the sake of harvesting these cells, which is the reason why there was so much controversy about pluripotent or embryonic stem cell research. But now there's a loophole. We can get all the benefits of the pluripotent stem cells with IPS, but bypass the ethical dilemmas. Usually to make our IPS stem cells, we're going to harvest some skin or blood from an adult, and then you have custom-made stem cells that perfectly match the genome of the patient that is receiving those stem cells. So we also are going to have no donor rejection issues. It's really pretty awesome. And there's some really great therapies that are being developed right now based off of induced pluripotent stem cells. So I was hoping to get to this review question on Tuesday. Let's do it right now. Please click on the gland, which uses holocrine secretions. The holocrine method of secretions. You're going to want to flip back several slides. We talked about three methods of secretion during class. We had apocrine, merocrine, and holocrine. Please click on the gland, which uses holocrine secretions. Make sure you submit your answers. Time is nearly up. Hit submit. So, guys, gals, as we look at this, let's back up and think. What is a holocrine secretion? We talked about three different secretion types. One is going to have budding. One is going to have secretion or squirting. And the other one is going to have the cells disintegrating. Holocrine is going to be associated with cells disintegrating. And of this, it is the sebaceous glands that are associated with cells disintegrating. Moving on, let's talk about tissue repair. When we have an owie, you get a cut, some kind of a damage that has incurred to your body tissues, there are two things that we do. We can have regeneration, and we can have fibrosis. The younger you are, the more regeneration you have. As we age, we have less and less regeneration and more fibrosis. So what's regeneration mean? When we have regeneration, we are going to reform the tissue, and we're going to have no loss of function. So we're going to replace it with the same thing. So if we lose some skin cells, those skin cells will be replaced with identical skin cells. There'll be no loss of function after healing. On the other hand, fibrosis, which happens more and more as we age, is going to be the replacement of lost tissue with non-functional scar tissue. Typically, it's going to be a dense, irregular connective tissue. Fibrosis typically will happen because we lose multipotent stem cells as we age. When you're fresh out of the womb, you have a ton of multipotent stem cells in your body. So you can have lots of regeneration, but as we get older and older and older, we lose our multipotent or adult stem cells, and it becomes more difficult for us to regenerate our damaged tissues, and consequently, we'll have fibrosis instead. If you get a cut to some of your epithelial tissues or damage to your epithelial tissues, there's some classic stages of healing that we need to talk about. Stage number 1 is going to be bleeding and clotting. So if we have damage occur, so the damage goes down into a blood vessel, that blood will seep into the wound, and as our wound is starting the process of healing, we're going to have some chemicals released. For instance, a kind of white blood cell called a mast cell releases histamine. Histamine is going to make our capillaries more permeable so that more blood plasma weeps into the wound area. As more blood plasma weeps into the wound, we'll have notably clotting proteins enter the wound site. And those clotting proteins help to plug the hole, stop us from losing liquids. And then the clot will form from those clotting proteins. Our blood clots are going to start to pull the edges of the wound back together. A superficial blood clot is known as a scab. A deep blood clot, a clot that did not break the surface of the skin, is oftentimes referred to as a bruise or a hematoma, to use the technical terminology. And as we're going through this process, we'll have other white blood cells, the big eater, the macrophage, move in and consume or phagocytize cellular debris that doesn't belong in that site of injury. Oh, here we go. Stage 3 of tissue repair from getting a cut is that we're going to have the generation of new blood vessels. So we'll have a neovascular, or new blood vessel supply being delivered, and those blood capillaries are going to regrow into the site of injury. Those capillaries, as they regrow into the site of injury, are going to be delivering lots of nutrients. We need lots of nutrients to be delivered because we're going to be having lots of cellular reproduction. In particular, epithelial tissues have a very high ability to regenerate themselves. And that's good because our epithelial tissues are going to be damaged continually throughout our entire lifespan. This process is going to begin typically 3 to 4 days after the injury has occurred, and depending on severity of the injury, it could be a couple weeks until we have regenerated the lost tissues. Finally, after, we'll say 2 weeks, we'll err on the side of a little bit conservative here. After 2 weeks of time, we have regenerated the lost tissues. So the stratum basale the epithelial cells from the stratum basale have repopulated and filled in the wound. And then down within the dermis, we have fibroblasts that have generated new collagen fibers or new scar tissue for that. And we have remodeled. We have maturated our wound. And this process will start from 2 weeks and can last up to 2 years of time. You probably noticed that, when you were a little kid, you skinned your knee, you cut your hand on something, you had some kind of an injury, and for a while there was a scar. But most of those scars you had while you were a young child are gone now. You don't see them anymore because that remodeling has taken place over several years, and that scar will slowly fade over time. When we need to get rid of tissues in our bodies, we can maybe have just the tissue dying from an unwanted cause. That is typically going to be known as necrosis, or we could have the tissue dying from lack of use, such as atrophy. So there's multiple ways tissues can regress, or the cells of the tissues can die off. If we don't use the cell and we have the tissue when we've lost of tissue mass from lack of use, we are going to have atrophy. And it's worth emphasizing that we can have a loss of size or the number of cells. It's very, very normal for us to lose neurons as we age. our brain's processing abilities typically will peak in the early 20s, typically about the ages 23 to 25 is when your brain is at peak processing capacity. So right now, you're closer to peak processing than I am, which is kind of scary to me. I'm on the downhill side of that number. And as we age, we'll typically have loss of brain cells and increase loss of brain volume. That's a very normal, healthy part of this aging process. We also could have disuse atrophy, a classic example of skeletal muscle tissue. Use it or lose it. If you are a couch potato, your skeletal muscle cells get smaller. If you're on mandatory bed rest for a couple of weeks, your skeletal muscle cells will turn into fibrous scar tissue. And if you're a gym rat and work out frequently, your skeletal muscle tissue will have the opposite. You'll have hypertrophy or an increase in volume of your skeletal muscle cells. If we have tissues dying off because of some pathological or disease state, we can refer to that as necrosis. Necro is the root word meaning death. Think of a necromancer, somebody who brings dead people back from the life. There's the same root word with necro right here, necro meaning dead. And there's a couple ways we can have necrosis. We could have an infarction. When we talk about an infarction, that is going to be a loss of blood supply, completely cutting off blood supply, and that loss of blood supply killing a tissue. The classic example of an infarction is a myocardial infarction. And I think most of you have probably heard of that phrase, myocardial infarction, also known as a heart attack, where you have a plaque clog of blood vessel in the heart, one of the coronary arteries, and then the cardiac muscle tissue, the myocardium, starts to die off from lack of blood supply. We also could have gangrene. Gangrene isn't typically going to be associated with a sudden cutoff of blood supply. Usually the connotation with gangrene is there's some kind of a pathogen causing a loss of blood supply. So instead of lipids clogging your blood vessel, you're going to have a reduction in blood flow from pathogens or pathogenic causes. There are different kinds of gangrene that can happen. As the name implies, usually there's a green coloration associated with the tissue as it starts to decompose while it's on the individual's body. So what are the different kinds of gangrene? We could have a bed sore known as a decubitus ulcer. This kind of gangrene results from staying in one position for too long. You probably had bleacher blood from sitting in the bleachers and your legs go numb. If you were to sit in the bleachers, we'll say for a couple days at a time and not move, eventually you would get a big ulceration or a big sore on your butt or your gluteal region as you had a cut off of blood supply to that part of your body from the pressure. We could have dry gangrene. When somebody has diabetes, they have chronically elevated blood glucose levels. Those chronically elevated blood glucose levels cause there to be more free radicals in the bloodstream, and peripheral capillaries of the extremities, are oftentimes affected. So individuals with diabetes will oftentimes have their toes and their fingers lose sensation. And as you lose the ability to feel things with your feet, you'll step on things, you'll stub your toes, you'll damage your feet and not realize you're damaging your feet. There's a reason why it's good for it to hurt when you have a rock in your shoe. Because when you have a rock in your shoe and you keep stepping on it, it causes a lot of tissue damage to your foot. And individuals that lose sensation to their foot, aka diabetics, will oftentimes have out-of-control infections in their feet and have their feet or lower leg amputated as a result. We could have wet gangrene, which is when internal organs are digested by pathogens. So we have liquefaction of internal organs. The classic example of wet gangrene is Ebola infections, liquefying individuals from the inside out. And we could have gas gangrene. Typically, gas gangrene is going to be some kind of a bacterial infection of the abdominal pelvic cavity with a large amount of gas, typically hydrogen sulfide or methane gas, so there'll be bloating associated with that. We could also have the cell pre-programmed to die. That's known as apoptosis. Apoptosis is a good thing. It's when we want a cell to die. We could have cells die because they're precancerous, they're about to become a cancerous tumor, or maybe we want one of our cells to die because it's infected with a virus, and it's now been turned and reprogrammed into a factory that's pumping out other viron particles, so we selectively destroy those cells that are now working against us as a whole. Whatever the reason for the cell being destroyed, after it's destroyed, it needs to be processed. So it'll be phagocytized. It'll be consumed by macrophages, the big eater cell. Macro meaning big, phage for eating. Every day, we have several billion cells die in our bodies on purpose, and it's a good thing that we have that happen. Most of our precancerous cells will commit suicide, so to speak, will commit apoptosis before they ever turn into that cancerous tumor, which is great. It's the ones that don't do that that become the problems, and those will typically start later on in life. It's been found that there is a surface receptor protein called FAS, and if you bind a molecule to the FAS surface receptor protein, that's like hitting the self-destruct button on a cell, and you'll immediately induce apoptosis for that cell. Once FAS is activated, we're going to have endonuclease, endo meaning in, nuclease, ace meaning enzyme, nucleo referring to nucleotides. An endonuclease is going to be inside of the cell, and it's an enzyme designed to chop up the DNA. So once the FAS is activated, the genome becomes destroyed. And without any genome, the cell stops to function. So that's it for epithelial tissues. Let's move on to connective tissues. As we look at connective tissues, this is another big, broad category. A key thing I want to emphasize with connective tissues is there is a lot of extracellular matrix. With epithelial tissues, there was almost no extracellular matrix. With connective tissues, the cell or the tissue volume is usually going to be mostly extracellular matrix. What do our connective tissues do for us? Well, they connect stuff. More specifically, it's really kind of a generic category. Any tissue type that was hard to categorize typically is going to be put into the connective tissue category. This category is the most diverse category of all the tissue types. Sometimes we'll have loose connective tissues that are highly vascularized, looping many blood vessels. Other connective tissues have no blood vessels associated with them. So, more specifically, what do these connective tissues do for us? They help to bind structures together. We have tendons and ligaments binding bones together and muscles to bones. We can have support, particularly from the kinds of cartilage and bone tissue in our body. We could have physical protection. Think of the bones, particularly your cranium, protecting your brain, or your ribs, protecting the organs of your thoracic cavity. We could have immune protection. Our white blood cells, blood being a connective tissue, functions to help with fighting off foreign pathogens that are in our bodies. We could have movement, particularly our bones provide the leverage that our muscles act on. We store energy in fat, or adipose. We store calcium and phosphorus in our dense bone tissue. We can produce heat, particularly when we're newborns. There's a special kind of adipose in our bodies known as brown adipose associated with infants. And this brown adipose tissue is designed specifically to make heat, because infants don't have a lot of skeletal muscle mass and they can't generate heat the way that an adult does, or even a little kid, by running around constantly or moving their bodies. And then finally, we use connective tissues for transportation. We use our bloodstream to move nutrients and waste products throughout our bodies. So we have a lot of variety with our connective tissues. There's also a lot of different cell types associated with our connective tissues. So let's start talking about cell nomenclature. Once we can break down the root words, it's a lot more approachable. So if we have this term blast. I want you to think building. A blast is a kind of cell that's going to be associated with building something. In particular, a fibroblast is a cell that makes fibers of the extracellular matrix, usually protein-based fibers. I'd mentioned macrophages already. Those are the big eater, or the kind of white blood cell that is going to remove pathogens and large bits of debris from our body. We have leukocytes. Leuko is a root word meaning white. Cyte is a root word that means cell. Leukocyte literally means white cell in the root words. And it's our white blood cells. Two common kinds of leukocytes include the neutrophils, which are associated with bacterial infections, and lymphocytes, which are typically going to be associated with our adaptive immune system and will attack viral infections and bacterial infections. Another common kind of cell associated with our connective tissue is a plasma cell. A plasma cell is going to fill the blood plasma with antibodies. So when we think of a plasma cell, that's the specific kind of cell that makes the antibodies associated with our immune system. And then finally we have mast cells. These mast cells are going to be associated with leukocytes. It's a form or variation of a white blood cell, and it's going to be releasing a lot of chemicals, heparin and histamine, associated with managing blood clotting. And when our mast cells are malfunctioning, they are also going to be associated with allergic responses and allergic reactions. And I believe on the bottom of the screen we have adipocytes. There we go. The adipose cells. Adipocyte referring to an adipose cell. Adipo meaning adipose, cyte meaning cell. So, here's all those cells jumbled together. And if we had a really great microscope and really phenomenal microscope slides, you'd be able to easily view all of these in areolar connective tissue. All of the cell types we just talked about are commonly present within areolar connective tissue. So we have a lot of these cells moving around outside of the bloodstream, but they use the bloodstream to get from point A to point B. Also note, there's a lot of variation in these cell types of the connective tissues. We have a lot of variation of the tissue, which means we need to have a lot of variation of the cells that make up those tissues. Our connective tissues have lots of extracellular matrix. Within that extracellular matrix, we're going to have a lot of fibers. The three most common kinds of fibers are collagen, reticular, and elastic. Collagen is by far the most common. It's the single most common protein and fiber in our bodies. We are approximately 12% collagen by mass. For our dry mass, I should say. So if you remove all the water in our bodies, 12% of what's left over is going to be collagen, or approximately 25% of all of our protein is collagen. So it's a big deal. And as we look at the collagen, it's used to make up the extracellular matrix. Depending on the orientation of the fibers, we can have flexibility or we can have stretch resistance. It's all about fiber orientation. We could also have reticular fibers. And as we think of reticular fibers, I want you to think of filtration fibers. We typically are going to find reticular fibers in structures in our body that are filtering substances. And when I say filtering, I mean removing pieces of debris from fluid. So we'll have reticular fibers in our lymph nodes, and as our lymphatic fluid is flowing through our lymph nodes, the reticular fibers tend to catch bacterium and other pathogens that have been captured by the lymphatic system. We also have a lot of reticular fibers in the spleen, which has the one function of filtering out old, damaged, or defective erythrocytes, red blood cells. We also have a lot of elastic fibers. As their name implies, they're the stretchy ones. They're elastic. The protein that makes the elastic fiber is called elastin. For those of you who Spent a lot of quality time looking at the areolar slides in lab. You notice the elastic fibers in the areolar tissue, or those dark purple skinny fibers that tend to be really twisty and turny on the slide. And that allows for them to stretch out and recoil and having elastic property. We also have the non-fibrous component of the extracellular matrix. Connective tissues have a lot of ground substance. So just to review again, when we look at tissues, there's the cellular and the non-cellular component. The non-cellular component is extracellular matrix, and within the extracellular matrix, we have the fibers and the ground substance. This ground substance is usually going to be fairly liquidy. Sometimes it's a complete liquid, sometimes it's more kind of goopy, and sometimes it's very stiff and rubbery. Depending on the tissue type, the ground substance can have a different consistency. If we have a gelatinous or rubbery consistency, there's a special kind of glycoprotein called a glycosaminoglycan. And these glycosaminoglycans are designed to have water molecules stick to them. They are a polysaccharide that have some amino-based sugars, and they are going to allow for our tissues to absorb water at a molecular level. You can think of them as a molecular sponge. And of these glycosaminoglycans, which is kind of an umbrella term, the single most common glycosaminoglycan is chondroitin sulfate, which gives us the stiffness of our cartilage in our bodies. This has particularly led to the dietary trend of individuals that have arthritis will oftentimes take a glucosamine chondroitin tablet every single day with the hopes that they'll rebuild the extracellular matrix of the cartilage in the joints that's being defected or being worn down where they have arthritis. Other glycosaminoglycans include heparin and hydroluronic acid, but those are the minor ones. The one I really want you to focus on is the chondroitin sulfate. Within our connective tissues, we have two broad categories, the loose and the dense. Loose is going to have mostly ground substance in its extracellular matrix. Dense connective tissues are going to have mostly fibers within its extracellular matrix. All these fibers are made of proteins. That's why I keep slipping up when I say that. So protein-based fibers, the collagen, the elastin, and the reticular fibers of the extracellular matrix. Some classic examples of loose connective tissues include areolar or reticular, and then we think of the dense connective tissues. Those dense connective tissues can have the fibers all parallel with each other. Those are going to be dense regular connective tissues with parallel protein fibers. If the protein fibers are oriented in many different directions, we call it a dense irregular connective tissue. As we look at our areolar tissue, this is all over our bodies. Areolar tissue is very widespread throughout our bodies. And it's going to have lots of collagen and elastic fibers, a lot of blood vessels. So when there's kind of the miscellaneous space in our bodies, oftentimes that miscellaneous space is going to be filled with areolar tissue. You can think of the areolar tissue as kind of like packing material. If you just need to fill a cavity in our body with some tissues to help bind everything together, oftentimes that tissue is going to be areolar tissue. So connecting our skin to the underlying muscle, we have a bunch of areolar tissue within the hypodermis. Within our areolar tissue, the fibers are going to be going in random directions. Let's talk structure and function. This is a key idea in anatomy and physiology. Because the structure has the fibers going in random directions, you can grab your skin and pull it in multiple directions, and it can resist tensional forces in random directions because the fibers are oriented in random directions. Structure determines function. So we have areolar tissue, everywhere in our body. Very, very, very widespread. This areolar tissue is a classic tissue type that we use to supply nutrients to epithelial tissues. Epithelial tissues are avascular. They don't have any blood vessels. But right next to most epithelial tissues, we're going to have a bunch of areolar tissues that are going to have lots of extracellular matrix with lots of blood vessels supplying nutrients to that extracellular matrix, so the epithelial tissues, at least the basal layer or the bottom layer of cells, can have nutrient supply. So here's a photomicrograph of the areolar tissues. We can see that we have fibers in multiple directions. They are all over the place. We also have lots of nuclei from the fibroblasts. And more difficult to see in this slide, you kind of have to look in the background. You can see very large, out-of-focus collagen fibers, which are about 10 times the diameter of the elastic fibers. We also have reticular tissues. Reticular tissues, depending on if you're looking at a lymph node versus a spleen, are going to have different appearances. In particular, as we look at our reticular tissues, we are going to have lots of reticular fibers, and those reticular fibers are going to serve as a filtration screen or a sieve or a net that will pull solid materials out of the liquid and concentrate them in that location. I had mentioned the lymph node and the spleen. We as adults have lymph nodes and spleen with lots of reticular fibers. If you're looking at a child, particularly young children, toddlers and newborn infants, they have a very large thymus. We as adults have a little shriveled up chunk of scar tissue that used to be our thymus. And in particular, young children with lots of red bone marrow will also have a lot of reticular fibers. We as adults have had most of our red marrow replaced with yellow marrow, or fat, on the inside of our bones. Let's look at a tendon and or ligament, also known as dense regular connective tissues. These dense regular connective tissues have fibers that are roughly parallel with each other. And because they're roughly parallel, we call it regular. Because the extracellular matrix is overwhelmingly fibers and not ground substance, we call it dense. So when we think of dense regular connective tissues, I want you to immediately think tendons and ligaments. Now, let's talk structure and function. If I back up a slide here, areolar tissues have the fibers oriented randomly and can withstand stretching forces in pretty much any random direction. Tendons and ligaments, on the other hand, have all of their fibers oriented in one direction. They're a one-trick pony. They're amazing at withstanding pulling forces along the orientation of the fiber. But anyone who's ever torn an ACL knows that just a few pounds of force perpendicular to the fibers snaps them all. Then we have dense, irregular connective tissues. Our dense, irregular connective tissues are going to have mostly fibers in the extracellular matrix, but those fibers are going to be oriented in many different directions. The classic example in our bodies of where to find, give me one, I need to tie my shoe here, of where to find dense, irregular connective tissues is going to be the dermis of the skin. So in lab, when you're looking at your skin slides, you should have noticed how there's this, it looks kind of like hamburger, for lack of a better analogy. And you can see that there's fibers oriented in many different directions, and those chunks of fibers are very close to each other. There's not a lot of clear ground substance within the extracellular matrix. So let's pause for a moment and have another review question. Please match the tissue type with the location in the body. Match the tissue to the location. Since I didn't cover hyaline cartilage yet, and I shouldn't have put this one on the question, hyaline cartilage is going to be located in our ribs and articulated joints. My bad. But who knows, maybe you remember it from lab. Make sure to get your answers submitted. We're at T-10. Make sure to submit an answer to get your participation points. All right, and we are out of time. So let's go over the answers. I told you hyaline cartilage is associated with ribs and articulated joints. We just covered reticular tissues a few slides ago. Reticular tissues are commonly associated with the lymph node and the spleen. Dense, irregular connective tissue, I want to say, was the previous slide. That's going to be associated with the dermis of the skin. And then when we think of the stratified squamous epithelium, we covered that one in lecture on Tuesday of this week. That's multiple layers of latent cells. Oftentimes, that's going to be associated with the epidermis of our skin. So in terms of correct answers, this is what our correct answer is. And it looks like most of us were able to get most of the questions correct. So let's continue moving on with our discussion of connective tissues. Our next connective tissue is adipose tissue, aka fat, blubber, lard, pick your adjective. So when we think of this adipose tissue, its primary function is to store energy. But it does more than just storing energy. It's also going to be a filler. We have a ton of fat around our eyeballs. It helps to cushion our eyeballs to keep them from bouncing around in our orbital cavities. We have a ton of fat located by our kidneys, or if you look at the popliteal region in the back of your knee, lots of fat is located there. Fat is a good thing in moderation. So we need to have a certain minimum amount of fat in our bodies for protection, for insulation, and for energy storage. Healthy young ladies typically will have a fat percentage, body fat content of about 18 to 20% adipose tissue by mass in their bodies. And that's a good thing for a proper menstrual cycle or a functioning menstrual cycle. So I know this is a much maligned tissue type, but it's not that bad. We need some fat in our bodies. So we store energy there, and as we're looking at this fat, it does lots of great things for us. To recycle our fat, to process the fat, we need to put ourselves in a calorie, or I should say a glucose deficit situation. Our body is always going to preferentially use carbohydrates to generate ATP with cellular respiration, but if we start to run out of carbohydrates, either blood sugar or glycogen that's been stored in our livers, we then shift over to beta oxidation and start to mobilize fatty acids from the adipose tissue. So, as we think of our fat types, we as adults have white fat. Sometimes it's referred to as yellow fat. And this is going to be very, very common. This is what we looked at in lab. The analogy I like is chicken wire. So if you look at this particular tissue type in a photomicrograph at high resolution, either 100 or 400 times the magnification, it looks a lot like chicken wire, like there's a bunch of hexagons or octagons interlinked with each other. Maybe you could call it honeycomb. Brown fat is in very young children, and it functions primarily to make heat. The purpose of brown fat is to crank out ATP. And as the ATP synthase is spinning in the mitochondria, it generates frictional heat. And one ATP synthase all by itself doesn't generate that much frictional heat. But if you have several 100 trillion of those enzymes spinning in circles, that generates a notable amount of heat, and that's how the brown fat will heat young children. Up on the slide, we have white fat for your viewing pleasure. This particular white fat is from the hypodermis. So as we look at these cells, they look almost empty. And that large white space within the adipose tissue is going to be associated with a very large droplet of triglycerides, or fat, that's being stored in the center. And then the nucleus is that little tiny dark dot that's squished against the edge of the cell. So that clear space within the adipose tissue, That's all fat. And the fat, being hydrophobic, doesn't absorb stains very well, so it tends to allow light to go right through it. Let's talk cartilage. As we look at our cartilage, within our cartilage, there's two key cell types that I want to focus on. We have chondroblasts and chondrocytes. So when we talk about root words for cells, blast means to build. What does a chondroblast build or make? It makes cartilage. So when we think of chondro, I want you to think cartilage builder. For chondroblast, a cartilage builder. The chondroblasts are going to make the extracellular matrix of our cartilage. And then as the chondroblast, and it's kind of a morphing process here, we start off with the chondroblast, and the chondroblast is just building up extracellular matrix all around it. It's just constantly surrounding itself with extracellular matrix. And eventually, it's trapped. It's like building a fort that it can't get out of. And as it's completely encapsulated with the extracellular matrix, the chondroblast morphs into a chondrocyte. And there'll be just a little tiny pocket of space left over. That tiny pocket of space is the lacuna, and within that lacuna, we have the chondrocyte. We also have the perichondrium. That perichondrium, for lack of a better analogy, is just some dense, irregular connective tissue that surrounds the cartilage to help give some scaffolding and framework to the extracellular matrix. Notably, the perichondrium contains extrachondroblasts. Contrary to popular belief, we can regrow and regenerate cartilage throughout our lifetime. It's just not very quick because our cartilage cells are typically going to be avascular. They are without blood vessels. So, well, yes, we have extrachondroblasts in reserve. Those chondroblasts have a supply chain issue. They aren't having lots of nutrients delivered in a timely fashion because the cartilage itself is avascular. It's without the nutrient delivery system. So cartilage tends to heal very slowly. But there are some things that we can do to speed up the rate of healing of cartilage. When we think of our cartilage, if we compress and squeeze the cartilage, we cause water or liquids to be released from it. And then when you relax the cartilage, it reabsorbs those liquids again. You probably noticed that if you've ever taken the time to measure your height in the morning and measure your height at nighttime right before you go to bed. Everyone is about a centimeter taller in the morning after being horizontal all night, and their vertebral column expands as the cartilage absorbs fluid. And then after walking around all day, you're about a centimeter shorter because your vertebral column has been compressing and squirting out extracellular matrix or ground substance from the extracellular matrix. So when we look at this healing process of the cartilage, we typically want to have just a little bit of compression. Mild to moderate exercise causes us to squeeze the cartilage and relax the cartilage and maximizes the rate of diffusion because we maximize the rate of fluid exchange with the extracellular matrix of the cartilage. Of our three cartilage types, hyaline, elastic, and fibrocartilage, hyaline is the most common cartilage type. Hyaline cartilage, for lack of a better analogy, I describe as eyeball soup or fisheye soup. Because to me, it looks like there's a bunch of eyeballs floating in some pink or purple or lavender soup, maybe periwinkle we'll call it. Now, within this hyaline cartilage, the extracellular matrix has almost no visible fibers. And because it has almost no visible fibers, it tends to be translucent extracellular matrix. And then the chondrocytes are going to be those cells with the nuclei within the clear spaces. Now, just because you can't see the fibers in the extracellular matrix doesn't mean they're not present. They're just too small to see with a light microscope. So they're very tiny. There's a variation of collagen that's very, very small, that's hard to see. Where do we find hyaline cartilage? We find it in any freely moving joint. We find hyaline cartilage around our rib cage, around the ribs. We find hyaline cartilage propping open our trachea and bronchi within our respiratory tracts. So we typically will find hyaline cartilage wherever we need a little bit of stiffness, but not a ton of stiffness. We also can have elastic cartilage. As the name implies, elastic cartilage has a lot of elastic fibers within its extracellular matrix. Elastic cartilage is typically going to be associated with structures that need some rigidity and a lot of flexibility all at the same time. So think of your external ear, the auricle of your ear. It's pretty dang flexible. You can grab your ear, stretch it out, wiggle it around, and then you let go, and it springs back into position because of the elastic cartilage associated with your external ear. Another key spot on our body where we have a lot of elastic cartilage is the epiglottis, that fold that covers up the opening to your trachea so that you swallow food and it goes to your esophagus and not your windpipe. And then we have fibrocartilage. Fibrocartilage, as its name implies, has mostly fibers in its extracellular matrix. It has a very high concentration of collagen fiber in its extracellular matrix. Now, as we look at fibrocartilage, the two spots in the body where we have most of our fibrocartilage include the intervertebral discs and the pubic symphysis. Intervertebral discs are going to be located in your vertebral column or spinal cord, and they're the shock absorbers in between vertebrae. When we think of the pubic symphysis, think of the pelvic girdle. You have two pubic bones in the front, and then there's a little bit of flexion you can have. And that small amount of flexion is associated with a chunk of fibrocartilage that separates the left pubic bone from the right pubic bone. And in particular, during pregnancy and birth, that fibrocartilage becomes even more flexible so the birth canal can open up and allow a child's head to pass through. And then finally, within our knees, we have shock absorbers built into our knees, extra chunks of cartilage called the menisci or meniscus. We have a medial and lateral meniscus for each of our knees. Or we have two pieces of fibrocartilage per knee acting as a secondary shock absorber. Let's move on and talk about our bones. There are two primary kinds of bone tissue that we're going to talk about, spongy bone and compact bone. And even within my lifetime, the names of these tissue types have changed a couple times, which is unfortunate. Because now I'm getting my brain bogged down with multiple terms that mean the same thing. So let's talk about spongy bone. Spongy bone is my favorite name for this because when you look at it on a microscope, it looks like a sponge up close, hence the name spongy bone. Another name for this is going to be trabecular bone. So if you're reading older literature and you see the phrase trabecular bone, the trabecula are the individual filaments that make up the spongy bone. So trabecular bone is the same thing, just with a different name emphasis. So those delicate strands are the trabeculae. And every once in a while, you'll hear this referred to as porous bone, but that's really fallen out of favor. Where do we find spongy bone? We find it inside of all flat bones and at the proximal and distal ends of all long bones on the inside. Emphasis on the inside. Spongy bone should never be on the surface. Or I should say spongy bone tissue should never be on the surface of bone. On the surface of our bones, we have compact bone tissue, sometimes referred to as dense bone or ground bone. So for your microscope slides in lab, they are labeled ground bone tissue, which is the same thing as compact or dense bone tissue. And when we look at the compact bone tissue, it doesn't have that spongy appearance to it. Instead, its extracellular matrix is going to be filled up with calcium-based crystals that give it a very rigid appearance. And we also have lots of tubes, particularly the central canals, that allow for a lot of blood vessels to move around within our compact bone. So here's a micrograph of compact bone tissue. Within the compact bone tissue, we have that osteon, that concentric ring structure formed around a dark central canal. That dark central canal has three things in it. It's going to have an arteriole, a venule, and a nerve. So it can deliver blood, it can remove blood, and it also is going to deliver sensation or nervous signals. That's why it's a little bit bigger compared to everything else. And then within the extracellular matrix of the bone, we're going to have, let's switch colors here to bright red, multiple layers around the central canal. Each layer or band around that central canal is referred to as a lamella because they are laminating themselves around the central canal. And then finally, as we're looking at this, we're going to have those little black dots on the edge. Those little black dots represent two things. They represent a cell and a space. The cell is an osteocyte, and the space is a lacuna. So within that osteon, I got ahead of myself here, we have the central canal, and then in between lamella, we have osteocytes. And those osteocytes are very difficult to differentiate from the space that they fit inside of. Chondrocytes and the lacuna that contains the chondrocyte usually will have some wiggle room, so the chondrocyte can move around a little bit. Part of the reason for this is as the tissue slide is prepared, we tend to dehydrate the tissue slightly, and as we dehydrate, particularly hyaline cartilage, the lacuna of the hyaline cartilage enlarges, and the chondrocyte of the hyaline cartilage shrivels up a little bit. So we can see a little clear space within the lacuna. But when we look at compact bone tissue, it doesn't shrivel up and shrink as it dehydrates. So the lacuna within compact bone tissue doesn't enlarge in the physical process of making it into a microscope slide. So they tend to be much tighter and smaller. And then as we're looking at the chondrocytes will have little extensions coming off of them. Those little tiny extensions are going to be going into a tiny space called the canaliculi. I had one student years ago, we referred to that as "can I lick your eye?" That's how he remembered to spell this correctly, whatever works for you. Those canaliculi are going to allow for adjacent lacuna within the compact bone tissues to connect to each other so that one osteocyte can communicate with another osteocyte. And then finally, around the surface of our compact bone tissue, wrapped around the perimeter of all bones in our bodies, we have a dense layer of fibrous connective tissues referred to as a periosteum. Peri meaning perimeter, oste referring to bone. So periosteum is a bunch of fibrous proteins wrapped around the perimeter or around the surface of a bone. It's also worth mentioning, in particular with our bone tissue, that it is the hardest tissue in the body. Bone tissue is the hardest tissue in the body. And then we have blood. Blood is one of the few tissues in our bodies that is a liquid at room temperature. A classic trick question is there a blood supply to the blood tissue? And I don't really have a good answer to that one. It all depends on how you frame your reference. I'll never ask you that question. Is blood tissue vascular or avascular? I don't know. So when we think of our blood tissue, the cells, we have multiple cell types. We have red blood cells, white blood cells, platelets. So we meet the criteria of multiple cell types working together. So it meets the definition of a tissue. But this tissue is special in that it has almost no protein at all in its extracellular matrix. And because it has almost no fibrous protein within its extracellular matrix, it has just the ground substance, and the ground substance can flow very freely. It's very liquidy. The blood plasma itself is the liquid at room temperature. Now, when I said red blood cell, the technical term for that is erythrocyte. For white blood cell, that's going to be the leukocyte. We will talk about these in more detail as the class goes on. The 5 leukocytes, the 5 broad categories are neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And if you have trouble remembering those, if you could just remember to never let monkeys eat bananas. That's how I remember the 5 categories of white blood cells. And then finally, we have platelets. When we think of our platelets, I want you to think of the cells associated with blood clotting. The technical name for a platelet is a thrombocyte. Let's move on to nervous and muscular tissue. So we're done with connective tissues, tissues that have mostly extracellular matrix making up the volume of the tissue type. When we talk about nervous and muscular tissues, these are electrically active tissue types. These tissue types can conduct electrical currents across their cell membranes. So they are excitable, and changing the voltage or changing the membrane potential is how they conduct electricity across the surface of their cell membrane. And when we think of this membrane potential, you could just think of it as a voltage. And it's not a very pronounced voltage. It's usually measured in millivolts. To give you some perspective, a AA battery is 1.5 volts, and you can pop that in your mouth and not even feel it. A 9-volt battery, if it's fresh and you stick it on your tongue, you can usually feel a little tinge from a 9-volt battery, but when we're looking at the millivolt range of the end of the spectrum, you never really notice it. Now, to differentiate nervous cells, nervous tissues from muscular cell types, let's focus on this electrical current and how it conducts electricity. In nervous tissue types, we're going to transmit signals to other cells, and we're just going to transmit the electrical information. Within muscular tissue types, the electrical signal is going to result in contraction or a shortening of the cell. So they can both transmit electricity. Only one of them gets shorter in response to an electric signal. The other one will just carry that electrical signal on to another cell type or tissue type. So let's focus on nervous tissue. When we look at our nervous tissue types, they function primarily for fast or rapid communication in the cell. Our nervous tissues are going to focus on both electrical and chemical information. The cell we typically care about is a neuron. It's the rock star. It is the most talked about, the most significant, and the most important cell type of the nervous tissues. That's the cell type that makes, propagates, and receives those electrical signals. And rightly so, we spend most of our time talking about neurons. But a neuron cannot do its job correctly if it doesn't have a supporting crew of cells. The support cells are the neuroglia, or sometimes just referred to as the glial cells. So these glial cells are going to be the support crew. So you can think of, like, if I think of like that wonderful rock band from the 80s, Bon Jovi. When most people think of Bon Jovi, they think of the lead singer, John Bon Jovi. And he's awesome. He's great. He makes wonderful music. But you know what? John Bon Jovi by himself is not that talented. You need the sound crew. You need the light crew. You need the bus drivers. You need the stage setup. You need the people handling the tickets. You need a whole crew of support people to give you a good concert experience. In the same token, or in the same analogy, a neuron by itself isn't really that effective. It really can't do that much all by itself. But if you take a neuron and combine it with a bunch of neuroglial cells, all of a sudden that neuron is awesome and amazing and does all those important things that we care about. It's worth noting that neuroglia are the most common kind of cell within nervous tissue by a wide margin. So if we look at this photomicrograph, All of these little dark dots represent nuclei of individual neuroglia cells, and then we have one nuclei of a neuron. So usually the number I hear thrown around is typically a 10-to-1 ratio of neuroglia to neurons within nervous tissue. Now within the nervous tissue, we focus on the neuron. The cell body of the neuron is going to be referred to as the neurosoma, or cell body of neuron. And then within that neuron, we have extensions coming off of the neurosoma. If that extension receives electrical signals, it's referred to as a dendrite. And if it sends electrical signals away, it is going to be an axon. And in particular, the axon of multipolar neurons can be very, very long. It's not unheard of to have a neuron be 1 meter in length, because that axon will travel from our brainstem all the way to the base of our spinal cord. Let's move on and talk about muscular tissue types. As we look at our muscular tissue types, the key thing that separates muscular tissues from any other tissue type is the ability to contract. This is the only tissue type designed to get shorter in response to an electrical signal. So what do we do with our muscular tissues? Overwhelmingly, we move stuff around. Either we move our bodies or we move substances within our bodies. So movement by exerting physical force is the name of the game of our muscular tissues. But they do more than just move our bodies and move stuff around within our bodies. Our muscular tissues also have a lot of frictional forces, as these actin and myosin filaments are sliding past each other within our muscular tissues. That generates a lot of friction, and that friction generates a lot of heat. That's why we tend to overheat and get sweaty when we exercise, because our muscular tissues are rapidly increasing the amount of heat they are generating. Of our 3 muscular tissue types, skeletal, cardiac, and smooth, there are some key differences I want to focus on. So when we look at skeletal muscle cells, skeletal muscle tissues, or skeletal muscle cells, are the most common kind in our bodies. And because skeletal muscle cells are very long, they can be 30 centimeters long, is not uncommon for a skeletal muscle cell. We oftentimes refer to them as fibers. And because they are so long, they need multiple nuclei along the entire length of the cell, so that one nuclei can control about 100 microns of the cell at a time. So as we look at a skeletal muscle cell under a photomicrograph, you can see nuclei all over the place. They are multinucleate. That's a key characteristic that differentiates skeletal muscle tissues from the other muscle tissue types. Another key characteristic is the stripes or striations. You can see the banded appearance within one muscular cell. Another key characteristic of skeletal muscle cells is we have the ability to voluntarily choose when to contract them. So we can choose when to send that signal to contract the skeletal muscle cells. You looked at skeletal muscle tissues in lab already. You have not looked at cardiomyocytes or cardiac muscle tissue in lab yet, though. Cardiomyocytes are notable in that they're only in the heart, and one muscular cell, one cardiomyocyte, can have multiple extensions coming off of it. So here I'll use green. We could have one cardiomyocyte giving us multiple branches. So these cardiomyocytes don't have that really long fibrous appearance as a skeletal muscle cell. When we look at cardiomyocytes, they can have 1 or 2 nuclei, so they aren't going to necessarily be multinucleate. Usually they're mononucleate. Something that's notable with cardiomyocytes is they have intercalated discs, which are represented by these dark bands that separate individual cardiomyocytes. And as I look up on the screen, I totally can't see it on the projector, but maybe on your individual laptop screens, you can see those darker bands that separate one cardiomyocyte from the adjacent cardiomyocyte. Those darker bands are referred to as intercalated discs. And those intercalated discs, to go back to Tuesday's material, are filled with gap junctions so that electrical signals from one cardiomyocyte will immediately jump to the cardiomyocyte next door. And because one cardiomyocyte can have lots of intercalated discs, can have 3 or 4 or 5 intercalated discs, if you electrify one heart muscle cell, all of the cardiomyocytes next door in an avalanche of contraction will start to contract. And this is good because when we start to contract our heart, we want all of the cells associated with one chamber of the heart to contract at the same time in a coordinated fashion. And then finally, we have smooth muscular tissue. Smooth muscular tissue is going to have a fusiform, or sometimes referred to as a spindle shape. Really, it just starts off at a point, gets wider in the middle, and then ends in a point. So as we look at it, they tend to have a shape that looks like this, with a nucleus in the center. Smooth muscle tissue does not have any striations. They're typically going to be associated with the walls of hollow organs. Think of the urinary system or the digestive system. That will have a lot of smooth muscle tissue. It's also worth emphasizing that smooth muscle tissue is not voluntary. We cannot control when it contracts. And to end our presentation on histology, what kind of muscular tissue contracts with the most force? I didn't mention this on the slides, but I want to emphasize it right now. The more striations there are, the more force it can contract with. More striations mean more force. So another way to reword this question is, what kind of muscular tissue has the most striations in it? And let's go with, because we're running out of time here, it's 9:15. I'll just put the answer up on the screen for us. The correct answer is skeletal muscle tissue. Skeletal muscle tissue. And that's all we have for today's presentation.